Great. All right, I think we're going to get started. Thanks everyone for coming. Today it is my great pleasure to introduce Marina Hutchins for her dissertation defense. Marina worked with Gil Rosenthal at Texas A&M on aggressiveness and sexual selection in a signaling context using fish before joining us here at Rice in 2019. Marina had like an advisor's dream project idea because she took the work that we had been doing trying to understand how aggression shapes social dynamics and considered it from a new perspective. So instead of just thinking about direct interactions between individuals, she had the insight that merely the opportunity to observe aggressive interactions could shape that individual's behavior and also the dynamics of the entire group. And so as you'll see in her talk, she leveraged ideas about social information, animal culture, and a variety of new conceptual perspectives to think about how social information can reshape social dynamics. And because it's flies, she did some clever experiments allowing us to pinpoint the role of specific social information transfer processes in conjunction with other important drivers, especially my favorite, genetic variation, to uncover new and important ways by which behaviors are and are not shaped by social information in genetically diverse populations. This approach has been very successful for Marina, to say the least. She has a first author paper in the American Naturalist, as well as another first author paper that's the first chapter of her dissertation that just got accepted to behavioral ecology and sociobiology, along with at least 2 more chapters in the advanced stages of preparation that I expect will be submitted soon to high impact journals and be important and valuable. Marina has won every award that our department offers that she's eligible for. Yeah. And when I put this in letters of recommendation, I always emphasize that we have a very strong graduate program with other excellent students, right? But Marina has still won all of these awards, along with external funding from the Animal Behavior Society, the Houston Livestock Show and Rodeo, and the prestigious National Science Foundation Graduate Research Fellowship. So not surprisingly, Marina was literally showered with postgraduate job opportunities, but ultimately accepted the competitive Stengelweiler postdoctoral fellowship at UT Austin with Molly Cummings, where she'll be working on these same ideas, but in a diverse population of fish. One of the key findings from Marina's dissertation is that a single individual can impact the behaviors of an entire group just by doing its own behaviors. And no one exemplifies this more than Marina herself. Marina has been an unstoppable force for diversity, equity, and inclusion in our department. She played a central role in the formation of our DEI committee, which she also served on for multiple years. She has served as a mentor for diverse trainees, from laboratory mentoring to high school and beyond, to more informal things like Skype a Scientist and a wide variety of outreach activities. And then there are the innumerable ways that Marina has impacted our community that can't be listed on the CV, including by just being the amazing and talented scientist that she is and being willing to be an example for other young scientists. It's been my honor and my pleasure to serve as Marina's PhD advisor. I've learned so much from you. And so now it is my job to turn over the microphone to you so we can hear about your research.
Great. Wow. Thank you, Julia, for that extremely kind introduction. Can everyone hear me okay here and in the Zoom? Perfect. Okay, well, hi, everyone. Thank you so much for being here. I'm so excited to talk to you guys about what I've been doing for the past 5 years in my thesis work that's been identifying the causes and effects of social information use and production using a fruit fly fight club. So the natural world around us is full of variation. Differences in trait expression at both the individual and the population levels actually allows for adaptations to changing environments. In fact, variation is the raw material through which natural selection can operate, thus leading to evolutionary change. An essential question then that biologists have been pursuing for centuries is, how can we understand the causes and the consequences of trait variation? If we're able to explain these patterns of trait variation that we observe, we can be better equipped to actually make predictions about how populations may change over time and their evolutionary trajectories. So to actually explain consistent variation in populations, scientists often turn to 3 explanations: genetic, environmental, or social. Let's look at birdsong, for example. The song that a bird produces could be the result of an individual's genetic makeup, or it could be explained by the location that we find the bird in, for example, if they're in a more urban environment. Or the songs that a bird produces could be explained by the songs that it hears from others. Importantly, these three explanations of variation are not mutually exclusive, meaning that it's almost never just genetic, just the environment, or just social factors that can explain the variation that we see. Thus, rather than view these as three distinct categories, we can now recognize that all of these sources likely contribute to trait variation. But exactly how each of these sources contribute to trait variation is not always clear. So I'm most interested in how we can explain variation in social decision-making. All of life is social. Even if you explicitly don't study social species or social behaviors, social interactions are important components of several events that happen over an individual's lifetime. Competition, cooperation, parenting, sexual selection, social learning, they all involve social interactions. So animals have this really complex social landscape that they have to navigate in order to be able to maximize their fitness. Thus, identifying how we can actually explain variation in animal social decision-making specifically is crucial to explaining patterns of animal behavior. So in order to make social decisions, animals can collect information by observing the actions of others. We refer to this as social information. And collecting social information can allow animals to reduce uncertainty, and it's typically a much safer behavior than direct interactions. We see social information across the animal kingdom. A very famous example is the honeybee waggle dance being performed by the bee that's sort of in the middle of that GIF there. So without even having to leave the hive, other bees can be informed about the closest food patch locations just by observing the behavior of the dancing bee. But humans use social information too. For example, if you see someone running down the street, what are you going to do in response? If you said run, first of all, you're correct. And second of all, you would be using social information in order to make that decision, because you can observe the behavior of another individual. And rather than stick around to find out why they're running, you can adopt the low risk behavior of just running as well. So let's refocus though back on social interactions and social information specifically. So there's two ways in which social information can actually impact social interactions. Firstly, individuals can use and extract information from the interactions and the signaling behaviors of others. This is commonly known as eavesdropping. Now, when I use the word eavesdropper, you might think of a hidden, shadowy figure who's hiding behind a wall and listening into conversations. But in natural social groups in the wild, this isn't the case. Rather, eavesdroppers are often able to be seen, heard by the interacting pair, and their presence can actually affect the production of social information. This is a phenomenon that's commonly referred to as the audience effect. So to understand variation in social decision-making, we can use this framework of social information in order to explain how genetic, environmental, and social factors all work together to influence social interactions. So we can manipulate the genotype of the individuals that are involved in this social decision-making, both the eavesdroppers and the interacting pair, in order to understand how genetic variation drives patterns and behaviors. We can also manipulate the social environment where the social interactions occur by adding an audience in order to understand how the presence of others can influence social information production. And finally, we can measure the responses of eavesdroppers to the social interactions that they observe in order to investigate if and how social interactions can cause long-lasting changes in behavior. So in my thesis work, I'm proposing to expand the conceptual framework and the empirical approaches for studying social information in populations, which will allow me to study the evolutionary feedbacks that are generated by social information use and production. I will accomplish this by addressing the following three aims. First, I'm going to characterize that relationship between social information use and production and its effect on individual behavior by answering the question of how eavesdroppers differ in their access to and use of social information. Then I'll investigate the genetic basis of social information use and production by measuring the genotypic correlations between the two traits. And finally, I'll explore how patterns of social information use and production can actually scale up to influence group level variation by answering if we can identify the transmission of social behaviors over time and how this process interacts together with genetic variation. Before we launch into the experimental aims, I'll give a brief overview of my experimental approach that I'll be using for all three chapters. In between aims 2 and 3, I'll actually present on one of the diversity, equity, and inclusion initiatives that I pursued as a graduate student in order to increase the participation of underrepresented student groups by conducting biological research with community college students. And lastly, if you fall asleep and forget everything I say, I will make sure to end the talk with some key takeaways to wrap us up. So I specifically study social information in an aggressive context. Aggression is a widespread behavior that many different animal species display, and it's important to be able to secure access to resources such as food, mates, or territory. Aggression often takes place in a social context where selection may favor eavesdropping in order to avoid costly interactions, and the presence of an audience can introduce new selection pressures to the interacting pair. Investigating how social information production and use can influence aggression, specifically in social decision-making, can allow us to create more relevant behavioral models when we're studying how aggression has actually evolved over time. Probably from the title of this talk, you will have figured out that I'm using fruit flies in order to answer my research questions. Fruit flies are great for this study questions because they form social groups on their food sources that may actually be stable over a number of days, both in nature and in the lab. And they provide the perfect setup for social interactions, which can be observed by many curious conspecifics. Fruit flies also have a well-studied suite of aggressive behaviors that have been well characterized. And we also know that fruit flies are able to learn from fights, at least the ones that they directly participate in. Fruit flies that lose a fight, for example, are more likely to lose subsequent fights in the future. Fruit flies are amenable to genetic investigations as there's a well-established set of quantitative genetic tools for them. And for the rest of this talk, when I refer to different genetic manipulations, I'm just talking about flies that have different genotypes. These are not flies that have had specific genetic mutations like curly wings or white eyes, for examples. These are flies that just have a known genetic background, and flies from two different genotypes are as related as picking two individuals at random in the population. Whereas flies from the same genotype have the exact same genes. And so they're essentially like identical twins. Being able to produce carbon copies of flies with the same genetic background allows us to easily investigate our questions about genetic variation while avoiding some of the conflicting results that can come from repeated behavioral testing on the same individuals. And finally, fruit flies have been shown to have the cognitive capacity for social information use in different social contexts. So for example, female fruit flies have been shown to copy the mate choice of another female, and they also have been shown to copy egg-laying sites. So thus, for all of these reasons, fruit flies are a very amenable study system in order to answer my questions about aggression, social information use, and genetic variation. So I designed an arena and an experimental protocol in order to test for how individuals varied both in social information production and use. This arena was composed of two main parts. The main part of the arena was two petri dishes with a small blue cap of food in the middle. This was topped with a bit of yeast paste, which is known to incite fly aggression. The observation tower was a pipette tip that was placed on top of the main arena and separated by only a thin layer of mesh. For my experimental design, I would insert two males into the main part of the arena. These males were dusted with powder to become orange and yellow to create two distinct phenotypes. This allowed for identification of the fly both for the human and the fly observers. I would then add an eavesdropper fly to the observation tower. And so this arena design allowed for information to flow between both the interacting pair and the eavesdropper. So auditory, olfactory, visual signals could all be passed between the two parties because of that thin mesh barrier that separated them. So after a brief acclimation period, I would measure all of the aggressive behaviors of the two flies in the main part of the arena for 30 minutes. This first half of the contest allowed me to quantify the effects of social information production, or how an audience affects aggressive behaviors. After the contest, I could then easily remove that observation tower and insert the eavesdropper fly into the main arena with the two male flies to interact with them directly. So this second half of the experiment tested for variation in social information use, or how males responded after watching an aggressive encounter. Yes, I am contractually obligated to make the joke that I am running a fruit fly flight club. I am trying to make this hashtag go viral. I will pay you a lot of money. If you know how to make that happen, please use it as often as you can. So now that we're familiar with my experimental approach, I want to talk about how we characterize the relationship between social information use and production in my first set of experiments. So we currently don't have a clear understanding of how eavesdropping and the audience effect actually interact with each other due to their characterization as two distinct and separate processes. However, by combining these two processes under the framework of social information, we can see that eavesdropping the audience effect may in fact form a feedback loop. So my first hypothesis for this chapter was simply a proof of concept. I predicted that fruit flies would be able to extract and influence information that's produced as a result of an aggressive encounter. I also predicted that changes in individual behavior are dependent upon modifications in the behaviors of others in this feedback loop. So a bidirectional information exchange here simply means that the interactions or the behaviors that the eavesdropper observes will change their own later behaviors, and the presence of the eavesdropper will influence the behaviors of the interacting pair. So my first chapter was a direct test of this hypothesized feedback loop. I had two main research questions. How does the presence and sex of an eavesdropper influence the production of social signals? And how do individuals actually then use social information to make decisions about how to navigate their social environment? Importantly, I tested both of these questions in a single experiment so that we could be better informed on how these two processes are linked. I had a total of 3 experimental treatments that I will walk through using this timeline. First is the male eavesdropper treatment. I started the trial by placing the interacting pair in the main arena and an eavesdropper in the observation tower. Then in the first half of the experiment, I recorded all of the aggressive behaviors between the interacting pair to assess how a male eavesdropper influenced the production of social information. And immediately after the conclusion of the contest, I inserted the male eavesdropper into the arena, and then I recorded the aggression of the male eavesdropper towards the interacting pair in order to quantify social information use. I also conducted a female eavesdropper treatment, which was identical to the male eavesdropper treatment, except in the first half of the experiment, I used a female fly, and then I recorded her mating decisions in the second half of the experiment. And my final treatment was a no eavesdropper treatment. So I left the observation tower empty in the first half of the experiment so that we can get a baseline for how that interacting pair would behave without an audience present. And in the second half of the experiment, I actually added a male or a female fly who was isolated, who had no prior social experience. Again, so we could see how flies would behave if they don't have access to social information. So for the results for this first project, like I mentioned, I conducted 3 different treatments, 166 experimental trials. I analyzed this data using generalized linear mixed models. So for our first set of results, when thinking about the effect of an audience on the interacting pair, I found that when a male eavesdropper was present, the interacting pair actually suppressed their aggression. So this is a box plot. On the x-axis is the identity of the eavesdropper that was present, and on the y-axis is the aggression of that interacting pair. You may also notice that the interacting pair didn't change their behavior whenever a female was present. In the second half of the experiment, I then measured how males responded after watching a fight, and I found that male eavesdroppers were less aggressive towards the interacting pair. So in this box plot on the x-axis is if the male was allowed to eavesdrop on the contest or not, and male aggression towards the interacting pair is on the y-axis. So again, after watching a fight, males lowered their aggression. Finally, when looking at our female data, we actually found that female eavesdroppers were not more likely to mate. They were not more likely to use aggression in order to choose who to mate with. This is a bar graph that just shows that frequency of how often females mated, if they had access to social information or not. On average, females did mate slightly more whenever they did have access to social information, but these differences in likelihood weren't significant according to those models that we created. So when you put all of these results together, we see support for the feedback loop in our male eavesdropper treatment. So as a reminder, on the social information production side, we found that the interacting pair decreased their aggression in the presence of an observing male. And on the social information use side, we found that male eavesdroppers also decreased their aggressive behaviors towards the interacting pair. Together, this indicates that males in the interacting pair may have been able to avoid elevated aggression from the male eavesdropper by adjusting and changing their signaling behaviors. Thus, our findings emphasize that access to social information matters for how male eavesdroppers change their behaviors, as variations in social information production led to differences in social information use. And on the female side, we found that the interacting pair did not change their aggression in the presence of a female. And on the social information use side, we found that female eavesdroppers actually didn't use social information in order to make their mating decisions. Together, these findings from our female data indicate support for the idea that changes in eavesdropper behavior are dependent upon modifications in the social information that's produced in their presence. In the female case, we didn't observe any changes in social information production, and thus we didn't observe any changes in how females actually use social information. So for this first chapter, we can see that social information use and production are linked processes that can help to explain variation in the social behavior of both the interacting pair and the eavesdroppers. Importantly, this confirmed our hypothesis that we do see this bidirectional information transfer between the social eavesdroppers and the interacting pair that has cascading effects on behavior. So we provided some of the first empirical evidence that's linked social information use and production in an aggressive context via a feedback loop. So with this finding in mind, we wanted to further probe this linkage by investigating the underlying genotypic correlations between these two traits, which could expose how these two traits could co-evolve. Specifically, we investigated plasticity in social information use and production. So a genotype who's very plastic in social information use or production will show a strong response to the social environment on their signaling behaviors. One that is aplastic will behave in a way that's consistent with or independent of any effects of the environment. This graph depicts two genotypes, one in green and one in black, that have two different responses across social contexts. The genotype represented by green shows more plasticity because they show that drastic change in behavior, whereas the genotype in black is less plastic. So theory predicts that the evolution of plastic behaviors should be favored in order to allow individuals to actually respond appropriately. Being able to adjust your behavior when socializing, depending on the social environment, is important in reducing conflict, maximizing your access to resources, and just surviving in order to reproduce. So using my previous experimental paradigm, we can quantify plasticity in social information use and production in an aggressive context. So for social information use plasticity, we can measure that aggressive response of the eavesdropper compared to a male who didn't get the opportunity to eavesdrop. Plastic genotypes will show a more significant shift in their behavior after watching a fight. And to quantify plasticity in social information production, we can focus on how a male in the signaling pair changes his behavior in response to different social contexts or different audiences observing him. Again, here's a graph depicting that green genotype that's very plastic, a black line depicting a less plastic genotype. So I hypothesized that selection should favor the evolution of correlated plasticity in these two traits because they relate to each other in meaningful and specific ways. Both of these plasticities require a response based on the same stimuli, being able to observe and respond to the actions of others. Thus, we might expect that there are shared mechanisms that underlie both of these variation in both of these behavior, or possibly historical selection that's linked these two traits together. So let's walk through some of the different relationships that we may see between these two plasticities. On the x-axis, we have plasticity in social information production. So as you move from left to right, genotypes become more plastic in response to being observed. And on the y-axis, we have plasticity in social information use. So as you move from bottom to top, genotypes become more plastic in response to observing a fight. So in these hypothetical results, each point corresponds to a tested genotype. A positive correlation here would indicate that genotypes that were most plastic in their production of social information were also most plastic in how they use social information, or vice versa. And from an evolutionary perspective, this would suggest that selection in any one part of the feedback loop could produce a correlated response in the other part. A negative covariance between these two plasticities would mean that genotypes that were most responsive to how they use social behaviors actually were consistent in their signaling behaviors, regardless of what audience members was watching them, or vice versa. And this could suggest the existence of a trade-off, or that there's specialists in the system. You can be really good at one of these two traits, but you can't be really good at both. And finally, no covariance in the system would indicate that there possibly isn't this existence of this unifying mechanism that connects the response to both producing and using social information. Instead, it would suggest that these traits could evolve independently of each other. So I had my main research question for this chapter, which was if there was a genetic correlation between plasticity and these two traits. But before we could identify a genetic correlation, we first just needed to identify if there's genetic variation in each of these trait plasticities. So those are my secondary research questions for this chapter. I tested a total of 14 different genotypes for plasticity in both social information use and production. So I expanded upon some of the genotypic variation that I measured in my first chapter. And I used the same experimental design, though, that I described previously. So to test for plasticity in social information production, I paired a focal male, who's indicated here with the red asterisk next to him, with a standard opponent. And then I recorded his aggressive response to an audience across 3 different treatments: male observer, female observer, and no observer. This allowed me to quantify how the focal male changed his aggressive behavior across the different social contexts. To test for plasticity in social information use, I had 2 treatments: the male eavesdropper treatment, where the focal male observed an aggressive contest, then was allowed to interact with those males directly, and the no eavesdropping treatment, where the focal male was isolated before interacting with the two males. These treatments allowed me to quantify how male aggression varied in response to social information. So I tested each genotype anywhere between 7 to 11 times for each of the treatments for a total of 980 trials across all 5 treatments. And again, for my data analysis, I built generalized linear mixed models. So to visualize the results for my social information production data, I have a large set of box plots. So I've broken the graph down into just one to walk you through how to interpret it before we see the whole thing together. So on the x-axis will be the genotypes. Each one is represented by a different letter. And on the y-axis is the aggression of the focal male. So each genotype has 3 different boxes that correspond to those 3 treatments for social information production plasticity. No observer, female observer in red, and blue for male observer. So when we look at this full graph together, my model indicated that we observed genetic variation in focal male aggression that was based on the presence and the identity of the audience. We see a number of different strategies that the genotypes adopted. For example, genotype H here showed an increase in aggression when they were being observed by an audience. Didn't matter if it was male or female. Genotype K here showed the opposite pattern. They were most aggressive when they were not being observed. So there's a lot of variation in this effect of an audience on aggression across different genotypes. For our data on plasticity and social information use, I'll be representing the data using this interaction plot. Again, I'm just showing one genotype right now so we can explain the full graph before we see all of the results. So in this interaction plot on the x-axis, we have if the males were allowed to eavesdrop or not, and we have the focal male aggression on the y-axis in the second half of the experiment. Each genotype is going to be represented by one of these lines that just show their average behavior of the males with and without social information. This genotype, for example, was less aggressive after watching that fight because we see that downward sloping line. So when we look at the full data set, we found that genotypes varied in their response to social information as well. Many genotypes tended to be less aggressive in response to watching a fight, which replicated results from my first experiment. However, we do see a few genotypes that actually increase their aggression after watching a fight, like genotype B that's represented by the orange line towards the top of the graph. We also see that the magnitudes of these slopes are different. So that means that some genotypes are more or less plastic. So now for the correlation between these two plasticities, we didn't find evidence of a significant correlation between plasticity slopes and social information use and production. So while theory suggests that these two processes can be linked by underlying mechanisms that allow for increased responsiveness across social contexts, our results suggest that that may not be the case. If there is an underlying mechanism, at the very least in our system, it doesn't vary with genotype. So future projects can continue to expand on this finding by identifying how factors, possibly other than genetic variation, can influence social information use and production plasticities in a variety of contexts. So what we can conclude from this from the second chapter is that genetic variation underlies plasticity and how individuals use and produce social information. This is the first empirical study that has investigated and linked genetic variation in these two trait plasticities, which is an exciting step. And with that, I actually want to shift into talking about my diversity, equity, and inclusion initiative. The students who actually participated in this initiative helped to collect data in my previous 2 chapters. And I included this diversity, equity, and inclusion initiative in my presentation and in my thesis, just to highlight some of my work and to show that a demonstrated commitment to DEI efforts will hopefully continue to be an area of emphasis and value in our department. And I also wanted to share the work I've done in this area to help generate new ideas of how we can tap into our existing community in order to accomplish our broader impacts. So historically, racial and ethnic minorities have been excluded from participation in many STEM fields, and this disparities in participation continue on into the present day. In the United States, representation of people of color is generally low across STEM fields, but it's actually exaggerated in our field. This bar chart is taken from a 2021 paper, which shows that in 2018, non-white students were more underrepresented among ecology, evolutionary biology, and conservation biology PhD recipients than in other STEM fields. One of the many ways to retain underrepresented students in STEM fields is by conducting undergraduate research. Jones notes in their 2010 paper that introducing students to undergraduate research, especially early on in their careers, benefits all students, but it may have disproportionate benefits for students from underrepresented groups. This is because undergraduate research can help students to integrate, both academically and socially, find systems of support, and secure their sense of belonging, especially while taking some of these STEM weed-out courses. Importantly, acting as a research mentor is a mutually beneficial relationship. Students, especially those from different backgrounds, can bring a fresh, diverse perspective to the research. As a mentor, I get the opportunity to hone my collaboration and my teaching skills. And one of the things that I benefited from the most is I get assistance from the students in performing experimental trials, data collection, and animal husbandry. So I created a program that would allow community college students to participate in biological research for my first and second chapters. The program I created also sought to help students actually navigate STEM careers. So to that end, I would review resumes and CVs. I hosted mock job interviews and mock graduate school interviews. With the desire to increase underrepresented group participation specifically in mind, I reached out to the local Houston Community College Network to recruit students for this program, where a vast majority of students are members of underrepresented groups. I was able to solicit more than 150 applications for this program. Over the course of 2 years, I was able to work with 4 of these students. And I'm happy to say that this program has very clearly been a success. Students were able to obtain Rice University research credits for their experience. They went on to present and were awarded for their work at conferences. Again, to my benefit, students assisted with data collections on 2 different sets of experiments. And importantly, for the goal of retention in STEM fields, all 4 of these students went on to be accepted at 4-year colleges into science majors after completion of the program. So I've reached out to each of these students who have graciously allowed me to share their updated information on what they're doing now. And I'm so happy to report that Beatrix is a senior biological science major at UH Clear Lake. Marcelo is a senior biology major at UT Dallas. Autumn actually graduated from here, Rice University, with a major in neuroscience and is currently working as a researcher. And Lean went on to become a senior biology major at Ohio State. So in conclusion, my DEI initiative provided opportunities for community college students to participate in research, which improved student retention outcomes in a mutually beneficial relationship. So to shift back into my research for my final chapter, we know that social information use and production are linked processes. They feed back on each other, and plasticity in both traits is determined by genetic variation and social context. So I then wanted to investigate how these processes could actually work together to scale up to influence group-level behavioral patterns. So the transmission of information via social learning can allow for behaviors to be spread in populations at a faster rate than genetic evolution. Importantly, social information production and use occurs simultaneously during the social learning events that are required for social transmission. Thus, in theory, I propose that over time, this feedback loop that I've identified can scale up in order to create patterns of behavior that are stable and that are explained via social interactions. The mechanisms through which social behaviors are actually transmitted via social learning, though, is unclear. This recent review in TREE highlighted the 2 primary ways that social interactions can influence group-level behaviors. Firstly, conformity. Individuals observe the behaviors of others and copy exactly what we see. This is what social learning often leads to. For example, monkeys learn to adopt the food preferences of their social groups via social conformity processes. However, social interactions can also drive patterns of differentiation, which is when an individual observes the behaviors of others and decides to adopt the opposite strategy. So for example, division of labor in highly social species like bees and wasps are regulated in part by social interactions. So our current gap in knowledge is predicting when we would expect conformity versus differentiation within groups. One hypothesis as to why individuals may choose to copy or to do something different emphasizes individual differences, as individuals can observe the exact same social information, but be impacted by it differently and thus choose to adopt different strategies. My entire thesis work focuses on how genetic variation specifically underlies individual trait variation. So I predicted that genetic variation would cause differences in these mechanisms of social transmission. So my research question for this final chapter addresses this gap in knowledge in social learning and genetic variation by asking how can genotypic differences in social information use and production scale up to explain group level variation in an aggressive context. I'm investigating this question by utilizing a transmission chain design. This experimental design is best explained by thinking of the game that is popularly known as telephone, but Julie and I are collecting data over all of the different regional names for this game. So please let us know what you may know this game as. If you're not familiar with it, it starts with one person who thinks of and whispers a phrase to the next person. That second person, who was initially the receiver, now becomes the signaler for the next person in line and continues this chain on to the next person and so forth and so forth. By the end of the game, you can see how much that phrase is true to the original utterance, or more likely has shifted and shaped it into something completely different due to the multiple steps of transmission in the process. So transmission chain experimental designs are really great for investigating social transmission. The role of social learning can be tested by seeding a behavior of interest into a group and then tracking how that behavior spreads from demonstrators to eavesdropping individuals. When those eavesdropping individuals become demonstrators at a later time point, transmission chains can actually simulate the process and the mechanisms through which social behaviors over time can become established and maintained in populations. In my experimental transmission chains, groups were seated with 2 demonstrator flies who performed an aggressive contest. And then over the course of 3 different time steps, these demonstrators were replaced by 2 other males for total group replacement at the 3rd time step. I had 2 treatments, eavesdropping and naive. In the male eavesdropper treatments, males were allowed to observe the contest. The focal males, again, are designated here with the red asterisk. These are the flies whose genotypes we manipulated and whose behavior we measured. So in step 1, the first focal male eavesdropper was allowed to watch a contest. And then in the second step, that eavesdropper replaced one of the demonstrator males. And another focal male was added as an eavesdropper. Thus, the first eavesdropper became the demonstrator in the second step. We repeated this process for the 3rd time step, replacing the remaining demonstrator male with a new focal male. And this last step, again, represented a total group replacement. So this replacement of the original demonstrators over time is what allowed us to test if social learning patterns in our system could scale up into group level behavioral processes by answering the question of if these males would continue to propagate the same behaviors in step 1, even after the original demonstrators were gone. In the naive treatment, males that were added into the experimental transmission chain were isolated and thus entering into the chain with no prior social experience. So in the first step, we took the isolated focal male, inserted him into the arena, replacing a demonstrator, and we repeated this process again for step 3. So this treatment allowed us to isolate the role of social learning in the propagation of aggressive behaviors. Specifically, we could answer if eavesdropping was responsible for behavioral patterns, or if direct interactions were enough and sufficient to explain the aggressive behavioral decisions that we observed in step 3. So to analyze this data, we built models that identified how the aggression from the focal males in step 3 of the experiment was predicted by both access to social information and the behavior of the 2 demonstrator males in step 1 of the experiment. We tested a total of 3 different genotypes and a total of 119 transmission chains. And for these results, I found that aggressive behaviors were propagated down the transmission chain by male eavesdroppers, but the mechanisms of this social transmission varied among genotypes. So these graphs show the total aggression of the demonstrator males in step 1 of the chain on the x-axis, and on the y-axis, we see the total aggression of the 2 males that were added to the chain at step 3. So the graphs are separated by the 3 different genotypes that I tested, and this blue line, again, is showing the male eavesdropper treatment. So within this eavesdropping treatment, we found that the effect of observing an increasingly aggressive encounter actually elicited different responses depending on the genotype. For instance, males from genotype A conformed their own aggression to match the intensity of the contest that they had observed, whereas males from genotype N show the opposite pattern. They differentiated by decreasing their aggression in response to watching an increasingly escalated aggressive encounter. So genotypes may differ in these strategies that they use in order to adjust their own behavior in a way that maximizes their genotype-specific fitness outcomes. So for example, a genotype that's really poor at fighting might benefit from differentiating their behavior and adopting the lesser aggression strategy, whereas a genotype that's really great at fighting might benefit from adopting the conformity behavior pattern that we see. So now I just added to these graphs the naive treatments, which are shown in the yellow lines. So in 2 out of our 3 genotypes, we don't see a significant relationship between step 1 and step 3 aggression when males were not allowed to observe before engaging in the transmission chain. This supports our hypothesis and indicates that social learning does play a key role in determining these group level behavioral outcomes and predicting what males will do at time step 3. In genotype A, however, Males showed the same behavioral conforming patterns even when they weren't able to watch the contest beforehand. This finding could suggest that for some genotypes, direct interactions are sufficient in order to explain behavioral patterns that we observed. As a reminder, all of these males got to interact with a demonstrator male in step 2 of the experiment, and this experience with a competitor alone could have been enough for them to adjust and shift their social signaling behaviors without needing to collect prior social information. So together, these results emphasize the importance of accounting for genetic diversity, especially when investigating social transmission events. So ultimately, from this chapter, we can conclude that this experiment adds to, or we can conclude that social information use and production, in conjunction with genetic variation, can help to explain patterns in social transmission. And this experiment adds to a small but a growing body of literature investigating the spread of social behaviors in populations, and to our knowledge, offers the first evidence of the social transmission of aggressive behavior via this experimental transmission chain design. So now's the time to wake back up if you were asleep. Takeaways. A1, we found that social information use and production are these linked processes that help to explain variation in the social behavior of the interacting pairs and the eavesdroppers. AIM2, we saw how genetic variation actually underlies plasticity and how individuals both use and produce social information. And in AIM3, we saw that these two processes, in conjunction with genetic variation, can scale up to explain variation in patterns of social transmission. So when we revisit this question in the beginning that I proposed of how trait variation, and specifically how variation in social decision-making, how we can explain it as biologists, I think I'm actually going to let our vice president summarize what we found. She does it pretty well. You exist in the context of all of which you live, the environment that you experience, the interactions that you see, and all of which came before you, your DNA, what makes you you. As predicted, genetic, environmental, and social factors, they all contribute to influence variation in social decision-making. My thesis work uncovered that it's important to move beyond simply characterizing the relative weights of each of these influences on individual variation, because these processes can all actually interact with each other, resulting in really interesting and complex patterns of behavior. So my thesis contributes to a better understanding of these interactions, leading us to become better equipped about how to make predictions about the behaviors of diverse populations over time that are teeming with genetic diversity. And as Julia mentioned in my introduction, I'm excited to continue this work. As a UT Stinglewire scholar, I will continue to explore this variation in social decision-making by actually investigating the ecological drivers of social information use and production in a sexual selection context using self-fed Molly. So I'm excited to do this work in collaboration with Molly Cummings as my next steps. And with that, we've come to my favorite part of the talk, the acknowledgements. It's not too often where you get to trap a group of people in a room and tell them all thank you, and I'm excited for this part. And I have a bunch of people to take. These acknowledgements are long, and I'm not sorry for it because I didn't just fall out of a coconut tree. I am the result of a community and groups of people that have been supporting me for a very long time. So first, I'd like to thank my Lord and Savior, Jesus Christ. Graduate school has been hard, and God's strength has allowed me to face a lot of the difficulties that come up in graduate school and really face them with this supernatural sense of joy and peace and patience throughout this entire process. To that end, I'd also like to thank my entire family who has come out in droves today for their continuous prayers and support. Shout out to the fam. My closest family, my mom, my dad, my sister, they've had a special role in really uplifting me throughout this journey. Even though they haven't always been able to summarize my dissertation research, they've never failed to show interest and unwavering belief in my scientific journey. I simply would not be here today if not for the many years that they spent unfailingly supporting a nerdy girl who loved science and animals. I'd also like to especially thank my husband, who is in the back rocking our baby, for his encouragement and his steadfastness throughout my time in graduate school. We got married right before graduate school. We're celebrating our 5-year anniversary this weekend. And he's really just seen me through this entire process. So Tim, you seem to always have the perfect thing to say whenever I needed encouragement the most. And you're always the model audience member during many, many hours of practice presentations. So I can't imagine having a better partner during this time. They say to never meet your heroes, but it's been an absolute honor to be mentored by my science hero, Julia Saltz. Julia, your excitement and your enthusiasm for my ideals long before they were worthy of a PhD has really motivated me throughout my journey. You've championed, you've advocated for me repeatedly, and having your support throughout my PhD has resulted in a body of work that I think is really interesting, rigorous, relevant, but it's also resulted in a thesis that's just been a joy to put together. To my committee, Scott, Corey, Simon on Zoom, your input has all been very valuable in shaping my thesis, and your endorsement of my work has really emboldened me as a scientist. I'm really thankful and grateful for your support at the many different junctions of my research project. I'm grateful to have been financially supported by several institutions that have provided funding for me over the years: the National Science Foundation, the Animal Behavior Society, Houston Livestock Show and Rodeo, the Biosciences Department, and the Rice University Institute of Biosciences and Bioengineering. Thank you to all of the past and current members of the SALTS Lab, as my work would not have been completed without the contributions of the lab, who are an amazing team of people that are always willing to lend a helping hand. A special shout out to Eric, Maddie, Charlotte, Anna, Leah, Patricia, and Gihan, who have all provided invaluable feedback on my experimental designs, read many drafts of my writing, provided experimental assistance, and just supported me in a variety of ways, both in and outside of the lab. I'm thankful to have worked with an amazing team of students, undergraduates, high school students, community college scholars, and exchange students, without whom it would have been literally impossible to complete all of the fly hours that are required to execute the over 2,300 experimental trials that I did over the course of my PhD. I reached out to these students to see what they're doing now. I'm so happy that so many of them updated me with their work. The very least that I could do is thank each of these students by name. So to Marcelo, Autumn, Anna, Leanne, Amanda, Sarita, Jasmine, Beatrix, Mahi, Tanya, Riley, and Anya. And to many other students who aren't pictured, I work with so many students, guys, but are worthy all the same: Jessica, Yukiko, Keshav, Aaron, Jason, Shoma, Kelly, Catherine, Becky, and Maggie. I'd like to thank all of these students for their incredible hard work and their dedication to my research program. There are many other colleagues in the Biosciences Department at Rice and beyond that I'd like to thank for their impact on me during my time in graduate school. But in particular, I'm especially grateful to Scott Solomon, who's on Zoom, Cassidy Johnson, and the team at Rice's Center for Teaching Excellence for encouraging my development as an instructor and providing me with valuable mentorship and opportunities to really practice the craft of teaching. I'm also really grateful for all the students, faculty, and staff that I served with and who continue to serve on the Biosciences Department DEI Committee. I'm also very thankful for Gil Rosenthal and Gaston Joffrey Rodriguez, who first introduced me to research as an undergraduate at A&M. They inspired me to pursue my doctorate degree, and they've continued to provide their advice, their insight, and their support long after my graduation. I'm so thankful for the camaraderie of the graduate students in my program and my friends who have seen me through my PhD. My friends have kept me smiling, and all of the students that I've befriended here have helped me to balance graduate school with a very rich social life. But I have to give a special shout out to my cohort who is here on Zoom today. Our cohort has been known by many names in the department. We prefer the very humble title of the best cohort ever. Oh, and Jaw's actually here in person today. And so Alex, Rob, Hunching, and Jaw, you all have provided me with invaluable friendship and solidarity that made graduate school really feel like a gift. My graduate school experience will forever be looked back on fondly because of the memories that we made. And finally, I'm dedicating this body of work to my son, Elijah. His timing into entering this world meant that we finished the last parts of this thesis and this defense together as a team. And I'm just so glad to have him with me. I wouldn't have had it any other way. Everything that I do now is for my son and for my family. So I'm so happy to dedicate this thesis work to him. And with that, I'm happy to take any questions.